My name is Desire Dalao. I am a genetics major, class of 2021, here at UC Irvine. I'm part of the Martin Lab, and today I will talk to you about the Viola sororia pigments. And yes, I think I'll have some time at the end for some Q&A as well. So there we go. Okay, so I want to start talking a little bit about me. So I've been interested in STEM classes since I was very young. And then I was able to find out that science could actually be used for something super cool, such as solving crimes. And this would also help me help other people, which I thought was really awesome. And after finding that out, I actually started developing an interest for DNA analysis, and that's why I'm a genetics major. I chose UC Irvine since it's a very beautiful campus, and I just felt really welcomed here and thought that it would be a great place for me to accomplish all my goals. So let's start off with a little background on plant pigments. So first off, what gives flowers their color? So anthocyanins are actually the most common pigments found in the chemical class known as flavonoids. Here you can see a difference in a chemical structure, which we're referring to are pigments, creates a difference in absorbance, which then creates a different color in the flower. Here we see kind of like a depiction of a flower gene with some a DNA sequence. This DNA sequence becomes a protein, which then helps make flower pigments. And what we will be changing is actually what these chemicals are made up of. And that will give us the different colors for our flowers. So the color of a flower can be very useful for them as it could help them with their survival and reproduction. This is as you can see, insects land on them and then are able to carry the pollen to other flowers and help them reproduce. So before I go into my data and all that, I wanted to go over a quick review of chemical structures, just in case some of you are unfamiliar. So as we know, carbon is a very important element that creates strong bonds, not only with itself, but with other elements. And this can form structures such as rings and chains. So here are some examples that you might have seen before, but there's actually a simpler way to depict the same thing. These are called skeletal structures. So if you see here, this is actually how you could also depict the same thing. So here, each corner or the ends of a line represent a carbon. Here, hydrogens are not shown, but we should know that there are enough hydrogens to make 4 bonds with a carbon. So you will be seeing some of these structures later on. So what is a pigment made up of? So pigments will actually contain what we call a backbone, which is the most common chemical structure that the anthocyanins will possess. So here we see this depiction of a backbone, and what will give us our different backbones will be what we call these R groups. R groups are just different elements, as you can see, or other chemicals. These chemicals will be added to these positions respectively. And based on what R groups are added, we will get a different backbone here. So we will be actually talking about dophenidine and cyanidine more specifically later. After you have your backbone, we have what are called add-ons or conjugations, which are then added to the main backbone to then form specific pigments. And these add-ons could be things such as sugars or acids. So why are pigments hard to study? So pigments have proven to be difficult to study with current techniques since they require large amounts of material to have good results. And some methods could also be quite lengthy. So here I will be talking about this paper further in the slides as well. But here we have this paper that here circled in red, we can see the different pigments that were found within just one flower. Here they were able to find this using 400 to 450 milligrams of flowers. With my technique, I actually used around 40 milligrams of flowers. So let's pretend I use these 3 flowers, which are, let's say, the 40 milligrams. So what they had to use was actually 10 times that. So imagine having to use 30 flowers for the same process. But I used a different technique that could only use the 40. So let's start with our sample preparation. So here I will start preparing. I'll show you how I prepared the sample before it was analyzed. So here first we obtain flowers and take their pictures. So these are the actual flowers that were sent to me all the way from New Jersey. I took some pictures of them before extracting them. So I obtained the flowers, took some pictures. Then you make sure to cut the tips off. These tips will then be weighed, and you always have to make sure to remember to write this in your notebook, as it's very important information. So here, if you can see, I'm not sure if you can see, but the scale actually says 41.4. So as I said, I used around 40 milligrams of the flowers for this. So now you put your petals in this Eppendorf tube, and you add what we call this extraction solution, which is made out of 5% formic acid and methanol. So the reason we use methanol when extracting is because this can get out all of the pigments within the flower, and not even the ones that are hydrophobic. So if we used water, it would not be able to get all pigments, since some would be not very compatible with water. So we want to use methanol in this step. Lastly, after 48 hours in the dark, we actually have to keep these in the dark, since pigments are very, they could sometimes break apart. So we want to make sure we keep them away from light for around 48 hours. We then pipette around 20 microliters of the extract, which was here, making sure we don't get any of the solid. We add the 20 microliters to 100 microliters of a 1% formic acid in water solution now. This will then give us a 10x dilution. So in this case, we actually use water once diluting, because this will actually help with our mass spec process. So after this, our sample is ready to be ran. So now I will talk a little bit about why pH actually causes a color change. So this is actually caused by changes in protonation state, whether your pigments will be conjugate acid or base. This will then affect how your anthocyanin interacts with what we call copigments, or colorless organic molecules in solution. So the copigment interactions are non-covalent, and this can lead to both of these molecules sharing a charge. This then changes what photons are actually absorbed by the molecules, and can therefore change what wavelengths are reflected, and therefore can change what we see. So here's like an example of some vials, different pHs, and we see how the color is changing here. So now I'm going to start talking about the actual technique we used. The first step here is what we call liquid chromatography. During this process, we will have two things, something we call the stationary phase and the mobile phase. This will be actually passing through a column. So this is an example of what we would see within the LC, which is this column filled with little beads. These beads, as you can see, pretend this is the edge of our bead. Our bead will have pores all around it, and we'll also have these very long carbon chains attached to them. So here we can see like a little zoomed in picture of what this group actually looks like. So this is actually going to be 18 carbons bound to a silica. So here we actually want long chains to have more interaction with what will be our sample. Here is the actual LC instrument. So we will actually have here on the top, these two bottles are what we call our solvents, or these are the mobile phase. Here, solvent A is 1.1% formic acid in water solution, while our solvent B is acetonitrile. So here, what we're gonna do is, as we change the concentration of our mobile phase, which as you can see here, this is a graph depicting, here's the percentage of our B solution. So let's pretend that if it's low B, that means we're gonna have more of the A solution. So here we will be changing from more A towards more B solution. This will cause our sample is going to be basically attached to the front of our column. And as we change the concentration, this will then start moving the molecules at different times, because this will be based on their hydrophobicity. So if the molecules don't like water, they'll start moving first and they will follow each other. And this will then allow further separation. So after the LC, we are getting prepared for the mass spectrometry analysis. So after this LC, our sample will be sprayed as a fine mist, which will be positively charged. So here we see, here is a little needle. And so through that needle is where our spray will actually come through. So here you can see this little picture. So our sample is coming out through here as a mist. And here we're gonna have the small particles with a positive charge. So the positive charge is really important for the mass spectrometer to be able to analyze these molecules. So this is a very important step. So after it is injected through here as a mist, it will actually go into the mass spectrometer. You might be able to see it here. This is the entrance to the mass spectrometer. So what will actually happen is our sample will be sprayed downwards, but the mass spectrometer entrance is actually on this side, as you can see here. So it will actually come down and go 90 degrees into the mass spectrometer. So now I will talk about the quadruple time of flight mass spec, which is now how we're going to determine the mass of our molecules. So the ions will be entering through here, depending on the voltage that is applied to these four poles, the quadruple rods, this will allow certain ions to cross, while the ones that are not matching the voltage needed will then crash and not be recorded. As this voltage changes, we will allow different ions to then cross successfully. So in this case, we will actually have smaller ions that will reach what we call the detector. They will be able to reach first. And larger ions will take a little longer to get there. And that's how we can determine the mass of the molecules crossing through here. So our results will end up being shown as spectral data, which will be shown in the next slides. So now we talk about the actual results. So these are the results of what I ran for the flower. So this is what we see as a typical spectral data of mass spec. So today we will be going over how I actually characterize or determine what molecule or what pigment is actually here. So this is actually the most abundant pigment found within the flower. So I will be talking to you about this now. So first off, we begin with the main peak, which is this one. What we're gonna do first, the first peak is gonna tell us what our backbone is. So what we do is we look at our mass And then I made a little possible backbone table down here. So we can see our mass is 303. So now we look at our table and we can see that the one that matches is this, which is our backbone, which we will say it's delphinidine. So now we have delphinidine here. I have circled it in red and will be circled in red in following slides just so you can follow along. So now we will go on to add constituents to our backbone now. So now to determine the constituents being added to this backbone, what we do is we look at the next peak. Here we see it's the 465. And what we're going to do is actually subtract 465 minus 303. This will give us the weight of our constituents. Our backbone was 303. Now we know that our backbone plus a constituent is now 465. So now what we will do is we subtract 465 minus 303, and we get this mass shift of around 162. So here's now our possible constituents table. I have included both the weight with H2O, water, or the weight without it. However, when characterizing these pigments, it's actually important to look at our weights without water, and I will explain why just momentarily. So here we see, so our shift is 162. We look for 162 on the weights without water, and we see it's glucoside. So what we do is now we have our backbone here, and now we will add a glucoside to it, which will be our first constituent. So we are actually not completely sure where this constituent actually adds on to our backbone, but based on the chemical properties, this would be the most likely place to be added. So now we move on to our next constituent. Now we will do the same. Now it's 757. We will now subtract this 465. That will give us a shift of 292. So this will determine what our next constituent is. We go back to our table, weights without water, go to 292, and here we go. It's a P coumaroyal rhamnose. So we will then now add it to our forming molecule. We still have here our backbone, our glucoside, and now the most likely place for this rhamnose to add would be right here. So here we're adding our second constituent. Now we have one more constituent to go. We do a 919 minus 757 now. We will see another 162 mass shift. So what we do is now we see 162 again, we see its glucoside, and we now add it to the most likely place, which would be right here. So now we have our backbone with 3 constituents added to it now. So now we can go here, and here you can see the flower I have analyzed, the Viola sororia. Here's our final molecule, what it looks like. And here's the data again. And then after we have formed this molecule, we can give it a name. So we can call this delphinidine 3O P coumaroyal rutinoside 5O glucoside. And now, as I mentioned before, there was another paper. There's a paper I found that actually did a similar study with different techniques, but they actually looked at Viola geruensis and Viola prionantha, and they were actually able to find the same pigment. As you can see, if you compare this data to ours, you can see that they have found the same pigment within these 2 flowers. So you can see how this pigment seems to be shared, at least so far, with the Viola family. So this last slide showed we talked about our main pigment. But now we can, if we look here, these are actually the spectral data of the following 2 most concentrated pigments within the flower. I have tentatively characterized these. These are not necessarily confirmed, but I did my best to try to come up with these. So these are actually the second two most concentrated, and these are actually have a cyanidine backbone. So that actually means this backbone is actually a reddish and a red color. And if we actually go back, our delphinidine is actually a blue colored backbone. Once we add the constituents, it actually starts turning a little violet. So that's kind of, it makes sense why our flower would be mainly violet. And we can see that there's actually red pigments that are within this flower as well. Sadly, our data did not suggest any yellow pigments within the flower. But it's cool that it seems like you guys found it. So I just want to end this by saying thank you for this project. This has definitely been fun. I had fun doing this for you. But this is certainly not the first time I have done this. While I have been in this research lab, I have actually helped with the analysis of over 100 flowers. And I have also helped characterize them, their pigments. This is all possible due to a list created by another undergraduate in my lab. So this list actually contains about, I think over 100 possible backbones. So if you remember from my previous slide, I had my table with possible backbones. So this is not the full story. This is just like 3 examples of possible backbones, but there's hundreds of them out there. For constituents, the same. We actually, here's a sample, just 3, but the list composed by the undergraduate is actually over 200. It has over 200 constituents in it. So that's actually what helps us characterize the pigments a little bit easier, but it's actually still difficult to characterize these pigments with accuracy, as some constituents actually share the same molecular weight. So unless other tests are used, it can be hard to actually determine what the actual constituent being added is. But yeah, I wanted to end with this, and here are my acknowledgements. Here are some members of my lab, and here are actually some members of the mass spectrometry facility, which is the facility I actually both work and do research here. So I want to thank them. Thank you.